Hello everyone. What we're going to talk about in this lab tutorial is the basic concept of microscopy. This is not an easy topic to talk about without being in the lab, but I'll do my best to cover the most generic information about it. So what we'll talk about here is, first of all, we go through an overview of microscopes. How do they work? What type of microscopes you have? And then we'll talk about the basic concept of microscopy. So the concept of resolution. What is that? The concept of magnification and how it relates to the size of what you're looking at, so called the field of view. And then we'll talk about the objective lenses, how some of them are defined as parfocal, and what is the working distance of an objective lens. And finally, we'll go through a few information on how you should properly label, draw first of all, and label anything that you see under a microscope. So, talking about microscopy in general, this is a very common technique. I guarantee you'll use it sooner or later in any lab you're going to be working in. And it's done to different types of studies. It's done to look at the morphology of different structures. This could be a sample, any type of samples, could be a full organism or a full sample, a slice, and so on. It's done to do actually live intracellular biology or biochemistry. We used to use it in the lab where I did my PhD, for example, to look at changes in calcium concentration. I put a video there, an example, a very cool video on how you can see the wave of calcium progressing through a Drosophila egg. It's not done in my lab. And then finally, it can be used to do diagnostic. When we talk about biopsies, so when you go for a biopsy in any hospital, that's exactly what they do usually. They just take it, your sample, they stain it, and they look if there is any variability and so on. And here you see some example. This is, for example, how you could detect prostate cancer. So this is how the tissue will look normally under a microscope. And then this is how it looks, infiltrated by cancer cells when you have prostate cancer. This is how it looks with inflammation. So by using microscopy, you can use it for different purposes. Here on the left, you have an example instead of how you can use it to visualize using fluorescence, different structure within the cell. In this case, this will be a fixed cell. You can see how the mitochondria have been stained using a red dye, or something that looks red under fluorescent microscopy. The microtubule fibers have been stained using a green dye, and here you can see where the DNA is aligned. This will be during metaphase of mitosis. We'll talk about that in the lecture. And this is finally another example of what it can be used for. This is a picture taken with an electron microscope, where you can actually get all the way to look at the size of molecules such as DNA. This is a DNA molecule. You can see actually the complexity of the molecule. So great potential for microscopy, can be used for different things. Now, there are different types of microscope. The one you'll be using in the lab is a basic light or compound microscope, like the one you see right here on the right side here. And here you have also a schematic of how an electron microscope will work or look or work. Now, no matter whether there are different types, you can have fluorescent microscopes and so on. They all work following some very basic common principle. First of all, they all have some sort of radiation or electrons. Electromagnetic radiation, for example, is the visible light or the fluorescent light that you can use to shine, or you can shine through a sample. An electron microscope will instead use electrons. You have a beam of electrons generated here, which is going to be shown through the sample. Together with that, then you would have a sample, a specimen, which is placed in what we call the stage, like this one right here, or this. And then the electrons hitting the specimen, or the electromagnetic radiation, the light, for example, hitting the specimen will create the image. This is a non-magnified image. So these electrons or the electromagnetic radiation will be scattered, will be diverted based on the density of the sample based on the orientation of the material in the sample. And so that's exactly what creates the image of the sample which is in the stage. And now in all electron microscope, or all microscopes, sorry, these images then get magnified. Can be magnified in different ways, using regular lenses, using mirrors, or using magnetic lenses like in electron microscope. And then once it's done with that, finally the magnified image reaches the final detector. In an electron microscope like this one, you would have two detectors which are connected to a camera, which then can finally take a picture of the magnified image. Or in the case of a light compound microscope, here you could have your eyes, or you can attach a camera, and so then that's where you get. So by doing this, basically you end up having a magnification happening of the original image, and therefore of the specimen, captured by a detector, whether it's your eyes or a camera. Now, obviously, depending on the type of microscope you have, you can achieve different results, particularly in terms of resolution, which I'll define in the next slide. Obviously, if you're using light microscopy, you can see only some very basic structures up to a certain size. We'll see on the next slide. So you have very limited resolution. However, obviously, the advantage is that they are very cheap type of microscope, so you can use them anywhere. If you use an electron microscope, the con is that it's very expensive to have one, but by using electrons you can actually greatly increase the resolution. You can see a comparison here. So this is exactly the same pollen particle there, which is taken a picture has been taken with a light microscope, and here with an electron microscope. You can see the huge difference in resolution you can get with the two of them. So what do we mean by resolution? Let's go through the first concept here, important for microscopy. Resolution is defined as the minimum distance by which two small objects near each other, adjacent to each other, can be seen as distinct. If you look here on the left side, that's an example that can apply to defining resolution of your eyes. For example, if you look sample 3 here, obviously you can see these two lines, or these other two lines, fairly well separated. Then the smaller you go, you start getting a bit of a hard time. I can still see a 6. I'm old enough. I'm old, but not that old. But then when I start getting to these samples here, I start having a very hard time to see them as separate. So this will be the distance of these objects here near each other, will be the resolution of my eye. Same thing for a microscope. So the resolution of the microscope is basically that smaller distance that can be seen, allowing to distinguish two objects as separate. And here you see a comparison in this table between the resolution of your eye, which can get down to a fraction of millimeters, so 0.1 millimeters. This is the size of a hair. A hair is a bit thicker than that, but close to that. With the light microscope, instead, you can go down all the way to basically 1,000 times less, almost to 100 nanometers. So you can see bacteria fairly well. You can see some picture here, some example. But you can't go any lower than that, whereas with electron microscopes, instead you can go down all the way to 10 to the minus 10 meters. So all the way to small molecules, such as this DNA molecule that I showed you before. Now, the resolution of a microscope is dependent on different factors. It's a complex thing. However, there are a few things. So one is the angular aperture of a lens. By angular aperture, you mean the half of the angle through which the light enters the lens on the objective lens in this example. It depends also on the medium that it's in between the object and the lens. So if there is air, it's a certain type of media. If you have water, it's different. If you have oil, some lenses indeed require oil to get the maximum resolution possible. So these as well reflects or slows down the traveling of the light, and therefore change the resolution. But the biggest factor is also the wavelength of the light. That's the biggest factor that affects this. So in general, the larger the wavelength, the larger is the resolution. And approximately you can calculate the resolution as half of the wavelength of light that you use. For example, if you're using visible light, now the maximum wavelength that you can use in terms of electromagnetic radiation is about 740 nanometers. So the range of resolution here can go up to about 350, 360 nanometers. So not far from the theoretical value of 0.1 micrometer. So you're in this range here, sorry, right here, which is what I was discussing, sorry, right here. So that's where you can get at pretty much. So let's start looking at some microscope parts so we know which part we're talking about, and let's discuss quickly what they do. Normally in a lab I will show you with a microscope right in front of you so you can actually play with the different parts. But this is the anatomy of a light microscope. That's what we'll focus on from now on, is the one you will be using in our own labs. So as I said before, it has a source of illumination, so you always have a light, in this case on the bottom. Now the intensity of the light can be adjusted with the brightness adjustment wheel here, and obviously can be turned on and off with a light switch. This is important to play with the amount of light. It helps with the overall resolution that you get and helps also not burning your eyes. Now once the light has been generated here, it has to go through a condenser. This is a lens that basically changes the angle or the width of the cone of light that's going to enter the sample. So you can make it wider or smaller, more focus. Might focus the light more in a single point. And this helps greatly with the resolution and the contrast that you get in the final image that you generate. Then you have a stage. This is where you will put your sample. Your sample would not be put as is, but will be first put onto a glass slide with a cover slip, which has the function to protect the objective lenses, and then the slide will be put on the stage, kept in firm using this metal clip right here. Now this stage can be moved. So basically this is where the sample is, the light goes through and creates the image, which then gets captured by the objective lenses. Each lens would have its own specific focus, and the focus is achieved by moving this stage up and down. How do you move this stage up and down? Is by using these adjustment knobs. So there are two types, one called fine adjustment knob and one other one called the coarse adjustment knob. The coarse adjustment knob moves the stage up and down faster. So basically it's done to do the coarse adjustment of the focus, hence the name coarse adjustment knob. Once you get close to focus, you may get an image in very close focus with the objective lens that you're using. You want to get the perfect focus, and to do that then you work with the small wheel here, which basically is moving this stage so slow that you can't even see it by naked eye, but it's still moving it so that you can get the real perfect focus. Once the image has been created here with the light going through the sample on the stage, now this image is captured by the objective lenses that are above it. The objective lenses are attached to what is called a nose piece, which can be rotated so you can interchange. They have different powers, we'll talk about that later on, different magnification powers, and they work at different distances from the sample on the stage. So they have to be perfectly aligned, so this nose piece has to bring them really at 90 degrees compared to the sample that is there. So the image will go through a first level of amplification through the objective lenses here, and then through a series of mirrors here will be directed to a second set of lenses, which are the ocular lenses. These lenses here can be one or two, in this case is two, you can use both eyes, and they have a fixed power. So in your case they would have a 10 times power fixed. So by doing that, now you have that the image gets amplified here, deflected, amplified even more here, and gets perceived by your eyes or a camera if you attach it here as a much larger image than what it really is on the stage. So first thing we'll talk about here is the objective lenses. So these are these lenses right here, which are attached to the nose piece. They are interchangeable with each other, and these are the lenses that do the first level of magnification. So the image enter through them when they're on focus and gets magnified to a certain power. The power of magnification is specified in each lens. So this would say 4x means the image will get 4 times larger, or be perceived as 4 times larger if it goes through this lens, 40 times on this one. Now the second level of magnification, as I said before, it's achieved through the ocular lens, which has a fixed power of 10 times in your microscopes. Now they can be interchanged using the nose piece, but they always have to end up perpendicular to the sample on the stage, and also at the correct working distance. They all have different working distances, so which is defined as the distance between the lens and the top of the specimen. So each lens would have its own specific working distance that brings it basically to focus. And these working distance tend to get smaller and smaller while the power of the objective lens grows to a point that you are almost touching. That's one of the problems with microscopy at the beginning, and that's the reason why you use a cover slip, that some lenses get so close that actually you can touch the sample and get dirty. Together with that, some lenses, and you'll see in the lab, I will show this in the lab to you, they're defined as parfocal. What we mean by parfocal lenses is lenses that once you switch from one to another one. So let's say you go to power with the 4-time lens first, and then you rotate, you put the 10-time lens, and you look for the focus again. Normally these lenses will require very minimal refocusing. So the image that you had originally 4 times, 40 times total, will be now almost or close to focus. We need minimal adjustment to go in focus. The same thing when you move from one other lens to another one. That's the meaning of parfocal. Are lenses that when you switch from one to the other one require minimal focus. So once you have the focus with the first one, the other ones will be basically almost on focus once you switch. Now let's talk about the concept of total magnification. Total magnification is defined as the ratio between the actual size of an object as it is on the stage and the way it's perceived by the detector. So it could be your eyes in this example here. So for example, something that is 1 millimeter here could be perceived by your eyes as 10 millimeters here. So yeah, 1 millimeter, 10 millimeters. So the total magnification will be 10 times in that case. You can actually predict also that magnification by knowing the power of the lenses. And the total magnification will be nothing else than the product of the magnification of each individual lens. For example, if you're using a 4-time objective lens here, and then your ocular lens is a 10 times magnifying lens here, now the composition, the total magnification you will achieve will be the product of the two lenses, so it will be 10 times 4, 40 times. So something that will be 1 millimeter here at this point will be perceived as 40 millimeters on your eyes. Now, depending on the magnification, you're looking at a smaller area in the field of view. By field of view, we mean that area, that space that you can see when you're looking inside the oculars. It looks like a circular area like this one surrounded by a black area. So this is the field of view. Now, the size of this field of view will get smaller and smaller. So the real size on the sample that you're looking at will get smaller and smaller the higher you increase the power. You can see an example here. So imagine you're starting here with your 4-time objective lens, and so you are with a 10 times ocular lens, you will be at 40 times total magnification. And let's assume that the diameter of the field of view you're looking at is 5 millimeters. If you think what you're doing when you are magnifying, moving to a higher power or a higher objective lens, what you're doing, you're actually looking at a smaller area in here. So when you're moving, let's say from 4 times to a 10 times objective lens, so 100 times total, what you're doing, you're focusing on an area which is 2.5 times smaller than the original field of view. So what you're looking at is actually a 2 millimeter area of what used to be in the original field of view. So while the power has gone up by 2.5 times from 4 to 10 times, the actual field of view would have gone down proportionally 2.5 times. So from 5 millimeters to 2.5. Same thing when you're moving to the highest power, at least in your microscope, going from a 10 times to a 40 time objective lens. There is no conflict of interest obviously in this image. You would have gone up an additional 4 times from 10 times to 40 times objective lens, so from 100 to 400 times total. Hence the field of view would have gone down by further by 4 times. So from 2 millimeters we would have gone down to 0.5 millimeter. So the total increase here would have been 10 times from the 4 objective lens to the 40 times of objective lens. And this, what you're looking at would have gone down 10 times as well. So from 5 millimeters to 0.5 millimeters. This is also the reason why when you use a microscope, again, it's a practical thing I will discuss in the lab, but it's good to know. It's very important to center your object before you move to a higher power. When you work in a lab with a microscope, this is exactly what you always do. You always start with the lowest magnification power. The reason being that this way you can really see a larger area in your specimen and find what you want to look at. But once you decide what you want to look at, you want to make sure that it has to be in the center of the field of view. The reason is that if it's outside, now you're magnifying the central part of the previous field of view. So if it's outside of that area, when you move up, you will not see it. Instead, if it's centered, it would actually be present. What you really want to look at will be magnified. This is a very common mistake done using microscope. It happens all the time. Oh, my sample has disappeared. No, it hasn't disappeared. You just focus. If this happens, practically speaking, you will go back to the lower power. So you will go back from 100 times to 40 times, and you will make sure that you recenter it, and then you go back to 100 times. Last thing I'm gonna discuss in this very quick tutorial is how you should properly keep track of what you see under a microscope. So how do you do proper biological drawings or labeling? Nowadays we take a lot of pictures. You'll be allowed to take pictures of your samples too. So these labeling strategies can apply also to pictures. Obviously the drawing is slightly different apart. So in general, important thing you gotta do, you always have to write as much information as possible regarding what you are drawing in there. So you would have to write obviously what you're looking at. You always write down who's doing the drawing, so you be yourself, when it was done, and obviously in our lab setting also your class and your section. Together with that, you should give information on what type of mount you're looking at. So if it's a fresh sample, it will be a wet mount, or it could be a fixed mount. So it depends. So a wet mount, it will be a fresh sample just done. You just basically take your sample, you might add some solution, could be water, or could be an isotonic solution to make it last a bit longer, and then you immediately put a cover slip, you look at it. That will be a wet mount. Together with that, you should also give information if any staining are used to increase the contrast, so to make a structure more visible. For example, you can use metal blue. It will stain nicely the nuclei, the cytoplasm a little bit less, and the membranes. So you have to give it information of what was added there as well, and the total magnification at which the drawing was made. So in this case, this would have been done at 400 times using a 4 times objective lens plus the 10 times ocular. Together with that, another big rule is you draw only what you can see. You don't draw what you expect to see. For example, this is a picture of a paramecium. It's actually an eukaryote, single cell eukaryote. And you expect to see some cilia that they use for movement and to feed themselves with. These cilia, sometimes you don't see them. You are expected to see them, but sometimes you just don't see them. The lens might be dirty, whatever. If you don't see them, you don't draw them. You draw only what you can see and what you can recognize. For example, here there is a lot of things in the cytoplasm. These dots that are put there, they should not be there. In fact, there is no labeling corresponding to them. So you should actually ignore this white stuff. They should not be there. So you draw what you can see and what you can recognize. And so here it is in terms of labeling. When it comes to labeling, you should always point to what you are labeling, and all the labelings should be done only on one side. You can choose left or right, but they all stay on one side. They should be organized on top of each other in a neat way here so they don't overlap. And then each labeling should be joined to the corresponding structure by a straight line, not an arrow. You want to use just a straight line that points to it and not an arrow, because you want to minimize the amount of structure in that drawing that are covered by the line. So the simplest line is a straight line, you use that. So for example, in this case, this cilia is pointing to the cilia. This is a contractile vehicle here that's written right here, and so on. So by doing that, in the end, you will basically create a drawing that someone else could easily interpret and use when they're looking at the same samples. Now, the same thing would apply, obviously, if you have a picture. You would print your picture, or you could do with your computer directly the labeling, but you will use the same strategy. All labelings should be on the side, not on top of the picture, and should be joined to the corresponding structure using a straight line. So, summarizing what we talked about here, we first went through an overview of microscope, very brief overview, the type of microscope you have. We focus mostly only on light and electron microscope, and we discussed what they are for, and overall how they work, all with a source of electromagnetic radiation or electrons and so on, creating an image and magnifying it. Then we talk about the concept of resolution, so which is the minimum distance between two objects that can be seen as separate. And so we briefly touch on the different parameters, particularly wavelength, how it affects the resolution of any microscope. Then we talked about magnification. So what magnification is, we defined it, and we show how you can actually calculate the theoretical magnification as a product of the power of the lenses used in the experiment. Then we talked about the objective lenses, how what it means for focal objective lenses and what is their working distance, and how overall we talked also about how the magnification has an inverse relationship with the size of the field of view. And finally, quickly touch also on how you will properly do some biological drawings.